program responsible for the CEMIT experimental station in Costa Rica, Veracruz, responsible for the development in the agricultural engineering area from Philip Morris, development manager at Syngenta Agro S.A.S.C.B., from portfolio manager Latin America North at Syngenta Agro, market for crop leader Mexico at Syngenta Agro, and product development at technical support at hand. Please receive him with a big applause.
Thank you very much. The idea is to make a journey in the time with, from the prehistory, where the tools for agriculture was very bicycle. They use this kind of tools only to put out the weeds close to the crop they want. To the present, where we have high technologies, like, let me go back. Like a precision agriculture, I want to talk about it. Like GMOs, crops. It's not well accepted, the concept of GM crops, but I am not agree. I think that kind of technology is one very useful tool to reach the enough amount of food for the population we have in the world. So I want to start presenting presenting that the evidence of the first domestic crops was around 2,000 years ago. The first agricultural societies become in the valley of rivers, like the Mesopotamia, Egypt, and Indus Valley. There was close to the rivers in Nilo, Tigris, and Euphrates, and Indus River. After that, in China, I'm sorry, no movie. Yeah. There's it. 200 years ago in China, and after that, in Mexico, 1,500 years ago, the Olmeca culture in the rivers of Papaloapan and Coatzacoalcos. But all these cultures can have growing in the population and be successful because of the rivers was helping on it. In Latin America, the prehistoric American cultures use a technique of many crops in the same area. They planted corn, beans, squash in the same area. And if you go nowadays to the mountains of Chiapas, Oaxaca, and Guerrero, they are still using almost the same methodology that they used thousands of years ago. The only technology they use is one application on every side to kill the weeds present, and that's it. They continue with this methodology. This methodology have an agricultural explanation because they are putting in the same place gramínea crop like corn, and also the beans that can fix the nitrogen, so they have a kind of symbiosis. But let me tell you that the production of this methodology is around 800 kilograms of corn, that is the main per hectare, 10,000 square meters per hectare. And for example, Guanajuato State, with all the new technology, they are producing 17 tons per hectare. So it's 20 times more production with the technology that with this M.Sc technology that is good, but only for self-consumption. But when you are thinking in Mexico that you need to feed 120 million of people, this technology is not enough, never will be enough. Never be enough. Let me go fast in the history. In Greece, the major crops were wheat and barley, including peas, beans, and fava, and olives. In the Roman Empire, the original adopt the technology of Sumerians, and they also have emphasis in wheat. And they also export crops, import and export in the different regions they conquest. Now let me go fast to the Middle Age. The monastery spread through Europe, and become important center for collection of knowledge. With this knowledge, the agricultural knowledge was included. And during the medieval period, the Arab War was very critical to connect the technology between Europa, Asia, and Africa continents. They transport crops and they move technology, or they copy technology for the different regions. Becoming more recent years, in the 1700s, the mechanized seeding was invented. It was very important because they correct the depth and spacing of the seed in the soil. And what is the importance to have all the seed on the same depth and the same distance? It avoids, if you are planting the seeds in a correct way, you are avoiding interspecific competition. I'm going to show you later a picture of that. But this was a very important advance in technology. Also the plugs, I listened in the previous talk that the cultivate the soil is not very recommended. It is a zero-tillage technology. It's a well technology, but it's not for all the world applicable. It's not applicable for all the world. For example, the main crop, as I told you, was wheat. In the wheat, we eat the seed, the grains, and to take the grains out of the plant when the scale is big, it's a very heavy work. It's very difficult. They hit the grains against the wall, and there is very difficult. So they invented the well, this machine, stationary steam engine, machinery began to drive a threshing machine. This threshing machine first was moved with horses. After that, they invented in 1812, they invented a steam machine to move and separate the grain to the rest of the plant. This was also very important invention to feed all the population that was growing in the world. And the first tractor, I'm going to say, the first petrol-driven tractor was built in America at the 1890s. And it has a very revolutionary invention to produce more food for the growing population in the world. After the America's discovery, there was an interchange of crops like corn, potatoes, sweet potatoes, tomato, vanilla, cacao. Rubber tree was very important from America to Europe for the New World to the Old World, and from Europe of the Old World to New World, wheat, barley, rice, onion, apple, citrus, banana, come to America. The potato became a staple crop through the European by late 1700s. So the base of the food for Europe after that interchange these years was potato. And when we use the same soil for many times to produce food, the amount of nutrients is becoming down, and they are starting having problems of fertility. As in the previous talk, the doctor said that the natural fertilizer like mature of the coast is important. It's good. It's a good way to add nitrogen to the soil. Also, they found that it was not enough. They need to find another way to supply nitrogen to the soil. So they use the guano, that is excrement of seabirds and bats, and they import from Europe to Peru. But it was very expensive, and also this amount of guano has a limited amount. So with the time, the nitrogen supply becomes every year more important and more difficult. I'm sorry. So in 1798, Thomas Malthus wrote a theory that the population was increasing in an exponential way, and the production of food was increasing in a linear way. So there will be, according with Malthus, a moment when a catastrophe will come. So there will not enough food for the population. If you see, it was very early in 1798 when the people started taking care of the amount of food for the world. Let me tell you that at this moment, this point is not being reached yet because of the technology. And but there is still this problem nowadays. At these days, we have enough production of food, maybe not good distribution, but we have enough production. Let me tell you about this latest law. It's known as the law of the minimum. They say that the maximum potential of production, that is the genetical potential of one plant, is not reached because the yield is not controlled by the total amount of available resource, but by the limited resource. There is the disease, the abiotic factor, the pests, the weeds, the nutrition, and the more limited resource is controlling the amount of the yield. And I talked before about nitrogen. Nitrogen plays an important part of many essential functions of the plant, like chlorophyll, plant tissue, proteins, enzymes. And this picture are deficiencies of nitrogen this is in tomato, in beans, in corn, in grapes. The nitrate is absorbed. The nitrogen is absorbed in the way of nitrate of ammonium, in these two ways. And 78% of the air is nitrogen. But nitrogen is in the air, and the plants absorb the nitrogen in the soil. So there was a problem. We need nitrogen, we have a lot of nitrogen, but it is in the air. So in 1920s, this chemical from Germany received a Nobel Prize because they found a way to extract, to obtain from the air the nitrogen, adding hydrogen for water, for methane. So they apply pressure, temperature, and at the end of the process, they obtain ammonia. So two Nobel Prize was delivered for these two German chemistries because they found the way to obtain nitrogen from the air. The main nutrients for the plants are nitrogen, phosphorus, and potassium. Phosphorus and potassium are available in some kind of soils. So there is not so limited as nitrogen. The plants take up phosphorus as orthophosphate or oxygen and phosphorus. And it is a key element for photosynthesis, respiration, and biosynthesis of nucleic acids and membranes. In practical terms, the phosphorus helps mainly to have good roots in the plants. And potassium, it is part of more than 60 enzymes in the plant. It is very important in the stomach regulation, photosynthesis, water uptake, and in practical terms, when you have enough potassium, you have good flowers and good production of fruits. Talking about nutrition, we call macronutrients, as I mentioned, nitrogen, potassium, and phosphorus. Also, the plants need in larger quantities sulfur, magnesium, calcium, and also micronutrients are very important, but they are needed in lower quantities. Coming to nowadays, let me mention that precision agriculture technology is a way to have a sustainable agriculture. But the technology is basically to regulate by satellite the amount of, for example, fertilizer that the tractor is delivering depending on the need of the soil. So they map the soils with the amount of fertilizer that they have in certain areas, and it can be different in another area. So the amount of fertilizer that the machine is delivering depends on the needs of the soil. With that, the inversion is less. And for example, one of the biggest critics for the nitrogen, to add nitrogen to the soil for crop production is because of the excess of nitrogen can go to the subsoil, to the water and subsoil, and it can be toxic for humans. With this kind of technology, the amount of nitrogen will be enough for being absorbed, but not too much to be leaching in the subsoil. Let me tell you now about the irrigation systems. The most basic irrigation system is surface irrigation system. I would tell you that in average, the efficacy of this kind of irrigation is 60%. That means 40% of the water is going out of the reach of the root of the plant, or is losing in the channels. It's not in the area that we need it. After that, there is a sprinkler irrigation. With this kind of methodology to watering the plants, if we have it in a good design, that means we will have efficacies of 90%. And the basic idea in the sprinkler irrigation system is to have enough between the circumference of each sprinkler to have better uniformity doing translate overlap. Thank you very much. The overlap of the sprinklers, the amount of water in the soil. And the best way to irrigate or to water in the plants is a drip irrigation system. With this system, we have around 99% of, if it has a good design, we have 99% of efficacy. We have no losses in the transportation. So it is the best way to irrigate. But let me tell you, there are intensive crops and extensive crops. For intensive crops like vegetables, it's or production of fruit, like in tree fruits. It's easy to have this kind of systems. But for extensive crops like corn, like soybeans, like wheat, it's too expensive to have drip irrigation system. So they need to use this, and in some areas they don't have option to use this technology, and they need to use that. But the idea, even for extensive crops, is to move for this technology. But it needs a high amount of inversion. Not all the people have. Okay. And for example, in Mexico, we have around 20 million hectares of agricultural, and under irrigation is about 4 million, and the other 16 million are in rainy season. So all the water that they need becomes from the rain, but the production is lower. Let me mention the hydroponic system. I like a lot this kind of technology. There are crops without soil. They are in a growing media, and we need to supply nutrients, oxygen, and water through the pipes. Hydroponics have only what the plants need. No more fertilizer, no more than they need. And they have very high yields. Let me tell you, tomato on the hydroponic, a good hydroponic system, can have 2,000 or more tons per hectare, and in the field they are 40,000 production. So 5 to 10 times more in hydroponic system than in the soil. Here very close there is a park. They have it in some of the greenhouses, they have hydroponic systems. Talking about plantability for extensive crops. For example, if you have the same variety, the same management of all the crops, but the plantation have the right deep and the right space between the plants, all the plants have the opportunity to take the same amount of radiation from the sun, the same amount of water, the same amount of fertilizer, and you have ears in the same size. In this field, the yield estimated is around 11 tons per hectare, and in the same yield, in the same condition, but with the bad plantation, if you put different depths, the seed, and different distance, you have some stems that don't have enough growth. You have big ears and small ears, and the reduction in the yield is around 50%, only because of the distribution of the seed. So the technology is important if we want to have food for the population. Let me go back in the history. In 1840s, potato was the basic of the alimentation of the food in Europe, and they have a pandemic of this disease, Phytophthora infestans. This Phytophthora is more related with the blue algae than fungus. But anyway, they couldn't have enough production, and a lot of people died at that time. Not because of the lack of production, because they didn't know the disease and they didn't have the way to control the disease. So with this experience, we knew that we need to prevent loss to the pests, any kind of pests like disease, insects, weeds. And there are a lot of technologies trying to control it, like cultural controls. Cultural controls is like to rotate the crops. Biological controls is to use, for example, the insects to bite insects, to eat the other insects. Pesticides has been very criticized, but it's an indispensable tool if we want to have production nowadays. And quarantine laws, and pest eradication programs. No one is 100% satisfactory and universally applicable. Let me talk about pests. Pests for agriculture can be animals, fungi, plantas, chromista, that is a kind of algae, protozoa, bacterium, and virus. So we can have a lot of enemies. To develop new active ingredients of pesticides, of agrochemicals, they need to evaluate 15,000 new molecules to select only one. And they need to have 10 years of toxicological environmental study before to have the permission to commercialize it. Before a pesticide go to the market, they need to present a lot of information of this kind. It is only an example that they need to talk about the toxicological, oral, dermal, inhalatory, eyes, skin, sensitization, chronic toxicity, mutagenicity, carcinogenicity, neurological effects, reproductive toxicity, developmental toxicity, neurological effects, and the effect for the media. All these information, generate all this information costs thousands of millions of dollars, and it's necessary if they want to if the industry of agrochemicals want to commercialize only one product. And the patent expires on average 15 years after the moment of the patent. So you have maybe 20, no, in these 20 years you have only, you lose 10 years in environmental studies, so you just have 10 years to commercialize without generics in the market. So it's not very good business because we can have a lot of problems with the generics and the environmental studies. Let me talk about the pest insects. there are a lot of insects that there are no pests, but also there are a lot of insects that there are pests. The arthropoda, we call arthropoda the insect that the adults have six legs, let me tell you like that, and arachnida, they have 8 legs. So we divide by the kind of wings they have. For example, Lepidoptera, that means wings with the scales. Coleoptera, wings with scales. Orthoptera, and so on. So, for example, let me tell you that the Coleoptera, there is a species called Antonomus that they can, in a week, provoke that a complete production of a field complete of pepper production go down all the fruits only in 1 week. So it's very romantic to say I don't want chemicals in my food, but if in a large scale, if we want to have food, we need to control this kind of pests. Also, in arachnid kingdom, they have an agricultural pest that can be very dangerous. In this table are all the registered insecticides in the world. It is insecticide resistant action committee, and they have a classification by mode of action. It's not very clear, but the colors means the way that they are working. Pay attention of this one. It's a microbial disruptor of the midgut, the digestive system of the worms, because there is a BT, Bacillus thuringiensis, is a bacteria that causes in the only the Lepidoptera kingdom, it causes a kind of diarrhea. They kill Lepidoptera by diarrhea. And it is very important because we are going to talk GMOs, and most of the GMOs have the protein of this bacteria in the GMOs. So a lot of people say, oh, in GMOs we are eating nobody knows what. And we have been eating Bacillus thuringiensis since 50 years ago. Applied by tractor, not in the gen, but we have been eating it for many years. And thanks to Bacillus thuringiensis, we have food, and a special corn. So another important way to mode of action is the interruption of the nerve system and the movement, in general, the movement by nerve system or muscle system. Let me go. It is a scheme of the synapses for an insect, where all these points are synapses, and the different groups of insecticides have action in the different part of the synapses. For example, in the sodium channel, in the acetylcholine, that is part of the synapses, in the acid amino gamma butyric, in the receptor of the ryanodon, and in the crotogonal organ. Let me show you, for example, the ryanodon receptor modulators. If we go to the cell of the insect, there is the sarcoplasmatic reticle, and there is a lot of calcium that we need to release this calcium if we want, we or the insect, if we want to move a muscle, we need to release this calcium. So this valve, this opens when we need to release the calcium and close when we need to relax the muscle. This kind of insecticides are blocking this granulin receptor modulator, and the valve is open all the time, and the insect can move again. This group of insecticides was discovered for a plant. This plant is in the Amazon, Riana espaciosa. The Indians living, the people living in the Amazon saw that if they put the stem of this plant in the water, the fishes die and floating. They can eat the fishes. After that, with the studies, they saw that what happened was the valve was of granulin receptor was open and they can move. It is also this valve is very used in cardiac problems in humans. But in the case of insecticides, the use is to kill these kind of insects. For me, it's very interesting also this example of a cortoronal organ. If you remember, or if you know, the insects have exoskeleton. So all the structure of the insect is with the exoskeleton, and the cortoronal organ give the sensation, all the stimulus and sensation is in this part of the exoskeleton. The cortoronal organ is basically in the nerve cells, in the exoskeleton. So they perceive the stimulus, and they send the signal to the body to have the movement of the muscle. So some kind of insecticides block this sensation, the sensation of the stimulus, and that's the way they can kill the insect. So in this case, this pest received the insecticide, and this is like a drunk. He can control the movements. So they die because they can't continue feeding from the plants. The stilette is the way they use for alimentation. It is the stilette, becomes flaccid, and they can penetrate the plant again, and the movement is like a drunk, and they die. It dies. Another way to control the insects is blocking the synthesis of chitin. Chitin is a very important part of the formation of the exoskeletons. The insects, for growing, need to move the skeletons. They move many times in the life, depending on the species, but maybe 3-4 times in their life. So if the exoskeleton can be formed because they block the chitin synthesis, that means that the insect will die. It's called growing regulators. Let me go to the fungi. The fungi are very important pests for agricultural production. There are the bites by Ascomycetes, Deuteromycetes, and Basidiomycetes, and also the kingdom Chromista, Omycetes. There was considered fungi, there are no fungi, but the damage is similar at the fungus. I talked before the problem of in Ireland of the disease in potato. It was an omycete. Very fast, just to say that the fungi kingdom has cell walls with chitin, and membrane with ergosterol. And the Chromista kingdom have cell walls with cellulose. So the Committee of Action against fungal resistance, some of them have, for example, cell wall biosynthesis and sterol biosynthesis. That means that this kind of fungicides only work from fungus, but not for homicides, because they have different cell walls. Many agrochemicals works blocking the process of one enzyme with the active ingredients blocking the enzyme. Let me show you an example. There is a group in the Committee of Action of Fungicide Resistance, group 7, that causing inhibition of cellular respiration, blocking the enzyme succinate dehydrogenase. In the mitochondria, the part of the cells where the respiration is doing, there is the Krebs cycle, and part in the Krebs cycle is the succinate dehydrogenase, and this group of fungicides are blocking this enzyme. So the transportation of electron is blocked, and the fungus die because it can continue the respiration process. Another one is, for me, very interesting. There is a part of the fungus called actin. Actin are globular proteins that these proteins give a structure to the fungus cell. So the alpha is a part of the fungus that penetrates the tissue of the vegetables. So the alpha in the growing tip, in the growing tip, they have this kind of globular protein in microfilaments that give consistency to the alpha. So when the alpha is growing in the tissue of the plant, this actin is giving enough consistency to penetrate. After that, the rest of the organelles of the cell of the alpha is growing with the alpha. So when we apply a fungicide to disrupt the actin, we can see here with colorimetric photography, the actin are the green part in the tip of the alpha, and in the control with a fungicide, it is the form that we can see. The alpha, but when we apply this kind of fungicide, the actin is not working, so the alpha pretends to penetrate the tissue of the vegetable, and it breaks. And all the cytoplasms go out, so the tissue died. It is applied, the fungicide, you can see it loses the consistency in comparison with the no applied. Check. There are the tips of the alphas, and the appressories also with a good condition in comparison when we apply this disruption of the team. Go to the weeds. The widths are practically divided in dicotyledons and monocotyledons. Dicotyledons are the plants that grow when they emerge, they have two cotyledons. Monocotyledons, they have only one cotyledon to feed the plant when it is growing in the emergency. If we talk about dicotyledons, there are the groups of Amaranthaceae, Asteraceae, Euphorbaceae, Ruaceae, and others. There is a lot of species. The stem for wood when they are old. Branches grow. The row have branches. Leaves with reticular venation. There is the venation, and the leaves have this kind of development of the venation. The monocotyledons, more important, Poaceae. The dispersal units are seed, rhizomes, and others. They have fasciculated roots. The stems don't have secondary woody. Symphodile growth, that means that it grows in one main stem. And the leaves have parallel venation. A different of dicotyledon, this is a parallel venation. This is amount of herbicides, commercial herbicides we have. Let me show you some example of that. For example, the group of acetolactasa synthetase, they are blocking the formation, the synthesis of amino acids like isoleucine, valine, and leucine in the process of synthesis of these amino acids. Another interesting mode of action is the p-hydroxyphenyl pyruvate dehydrogenase. Is this enzyme that is blocked by the herbicide, and then when this is blocking the synthesis of this enzyme, this plastoquinone cofactor is not formed, and the plant can produce carotenoids. The carotenoids are very interesting photosynthetic pigments. Here I have chlorophyll A. Where is the spectrum of absorption? It's in the violet, in the blue, in the orange, and in the red. Chlorophyll B, similar. The spectrum of absorption of light is in the violet, in the blue, in orange, and red. So if the chlorophyll absorb violet, blue, orange, and red, why we saw the chlorophyll green? Yes, because it absorbs what we don't see, and it reflects what we see. What we see, the green. Excellent. And in the case of carotenes, it's a complementary. This is the carotenes. They absorb also the green, the blue, the violet, but they don't have any absorbance of yellow, orange, and red. So that's the reason why we know that the carotenes, like a carrot, are orange. Because if they don't absorb, they reflect. So that's what we see. So the carotenoids, as I told you, there are photosynthetic pigments that it helps to the chlorophyll to absorb the light and transmit this energy after that to the chlorophyll to make the photosynthesis. So as the carotenoids protect us for the light, also protect, or it was designed to protect the plants for the light. Also protect us for the lights. So we need to eat carotene. We don't have to have skin cancer. Because they are having the similar function that they have in the plant. Let me tell you a little about the physical-chemical properties of the pesticides. Very important concept is the octanol water partition coefficient. That this concept is the relation between the solubility in organic solvents like lipids and the solubility in water. What happens if a product has more solubility in organic solvents than in a water? This division will give us a big number, entire number. If it's more soluble in water than in solvents, it gives us a fraction. The logarithm of a fraction is negative. The logarithm of an entire number is positive. So when we have a negative octanol water partition coefficient, it means that it will be very mobile in the plant because it's moving with the water. If it's positive, it will mean it wouldn't be mobile in the plant because it can move with the water. So when a product can move with the water, it can move in upper way by the sealant, or another products can move in the phloem up and down. It is water and it is chlorophyll or biosynthesized. So when the product have acid reaction, it can move in the phloem. So if it is absorbing the leaf, it can go to the apex, it can go to the roots. If we want to control a weed that the growing point is behind the soil, we need an herbicide to move in the phloem, to go to the roots, because the growing point is in the roots. So the herbicides that if we want to control, for example, Cyperaceas, that this kind of weeds have the growing point behind the soil, we need this kind of systems. And all the information of the movement is in the physical-chemical properties. Let me go to technology of production of improving crops. Traditional breeding, the humanity has been doing traditional breeding since they have the wheat 10,000 years ago. Because the traditional breeding is to select the best plants and planting it again. It's a traditional breeding. After that, they can cruise, they can put the pollen from one plant to the female flower to make a cruise. Mutagenesis is another way, it's not very useful. RNA interference, it is interesting. Transgenics, a lot of information against transgenic. So let me tell you that at the moment, I don't see one scientific publication with base against transgenic. a lot of papers, but no scientific. And gene editing it's also very interesting. Gene editing is not moving the gen from one species to other. It's putting or exalting the gene we want. For example, I have this, that example. A moment. Okay, the traditional breeding is just simple. It's Sea perennis. It's an original plant, but the human was selecting and they produce Sea mice. Sea perennis and Sea mice have 99% of the genes similar. But we can eat this and we cannot eat this one. This was a selection of the people for many, many years. I have the honor to know this man. This man was Norman Borlaug. He got the Nobel Prize in 1970 because his research conducted in Mexico increased the yield of the wheat in 5 times. All using traditional breeding. They emasculate the wheat, they select which one to cross, and they improve the yield. It was called the Green Revolution. The work starts in 50s and 60s. They received the Nobel Prize in 1970. After that went war around, and in India, in the 60s, they have a Malthus very close, the Malthus prediction very close, because the population was growing and they don't have enough food for the Indian. Norman Borlaug went in the 60s to the Indian, and they obtained the rice iron 8 that increased also the production of wheat. Obviously, to have this increase, we need to increase the capability of the wheat, or no, in this case of the rice to produce, but also we need to feed the wheat, to fertilize, to give all the weight of the rice need to control the pests. Let me give you an example of breeding, or traditional breeding process. They select a plant that have a characteristic we want, for example, big ears, big grain, and it is a heterozygote. It has a lot of information. They select for 7th generation the plants with the information we want to select, and we have homozygosis. After the homozygosis, the inhybrid is very small here, and they have another selected plant, inhybrid B. When they cross these two, they have the hybrid viable. So they have a degeneration in the homozygosis process. When they have the result of this homozygosis and they cross, they have the plant we was looking for. It takes a lot of time, 7 generations. So after the time, the scientists found a way to reduce the time with duplication of chromosomes, with the haploid, with the haploid system, that is to provoke gen duplication. So they save a lot of time, causing more gen duplication. It's called the double haploid. I told you about the editing of the genes. Agrochemical company called Syngenta found the genome or decipherate the genome of rice. With this decipheration of genome of rice, they found that with modification of the genome, not trying, not bringing any gene for another plant, or just exalting a genome, they can produce a rice with beta carotene, a precursor of vitamin A, and with that beta carotene rice, a lot of people in Asia is not losing the sight. So it is by editing the genes. And the last, the GMOs. What is the GMOs? They cut a part of the genome of a plant, they insert a plant on another organism. For example, I told you about BTs, Bacillus thuringiensis insecticides. They put the gene of the BT that causes digestive problems to Lepidoptera, and in that way, the corn have protection about Lepidoptera. That is the main pest in corn. How they do it? In a very simple way. There is a bacteria called Agrobacterium tumefacis that produce it provokes tumors in the plants. So in one hand, they isolate the gene fragment they want, for example, the fragment of the BT that provokes diarrhea in the worms, and in the other hands, they have this agrobacteria that can provoke tumors. Tumor is a multiplication of tissues, right? So they insert in the agrobacteria the gene of BT, and they have now in the agrobacterium the BT. The BT was introduced in the tissue of the plant, and the tumor I told you is a reproduction, uncontrolled reproduction of cells. So the reproduction of the BT is in the plant. So that gene that is for a bacteria now is in the plant. And in that way, they can control the worms. But the shadow of Thomas Malthus continues. For the year 2050 in Mexico, now we are 180 million of inhabitants. In less than 30 years, we are going to be 156 million of inhabitants. Same in the world. This is the current population. For the year 2050, we are going to be 9,600 million of inhabitants in the world. So we need to continue improve technology if we want to feed all the population in the world. In my mind, Galileo need to face the inquisitors, because he says that the world move around the sun, and they don't understand that. If they don't understand that, they fight against that. GMOs are not understanding. So a lot of people fight against GMOs, like the Inquisition fight around to Galileo in this case, because they don't understand. Let me go back just to close the talk. Let me go back to show you again this table. I didn't talk about the transgenics. It's highly regulated and safety testing is required, like the agrochemicals. So no one GMO can go to the market if they don't prove that it's safe for the human, for the animal, for the environment. So that's why I think that the Greenpeace is like a inquisition. In my mind. That's it, thank you.
That's okay. Unfortunately, we don't have time for questions right now. Will you be able to stick around for lunch maybe? At one? Okay. Anyways, do you have contact information that you can share with us?
Yes.
Okay.
So we want to give you a present. Thank you so much for the class.
Thank you.